Great. So the first article I want to discuss is the article Engineering Values into Genetic Engineering, a proposed analytical framework for scientific social responsibility by Pamela Sankar and Mildred Cho. So one of the things that they point out quite early on is that the new techniques for genetic modification that CRISPR-Cas9 represents is renewing old debates. So as I mentioned a few slides back, these debates about the ethics of using CRISPR-Cas9 for genetic modification are not new. And they point out that what's sort of troubling about the debates around CRISPR-Cas9 is the fact that when scientists and ethicists react to a new technology and discuss it to form an ethical consensus about its use, the consensus doesn't generalize to other genetic modification techniques that scientists later develop. So in some ways, Sankar and Cho are accusing scientists and ethicists of reinventing the ethical wheel every time a new genetic modification technique comes along. And so one of the things that Sankar and Cho would like is for scientists to be able to consider the social and ethical, I think, by proxy, implications of their research as they develop their research agendas and protocols. And I think the idea here is that if scientists are attentive to these social and ethical issues as they're developing the technology, then you won't have these debates once it comes on the scene. There won't be a scramble to figure out how to react. I think this is a interesting move on Sankar and Cho's part. Excuse me. Fang Chang, who is credited with being one of the people who created the CRISPR-Cas9 technique, was also instrumental in the development of the TALENS technique for modifying genes. And so one wonders, had there been a process in place to consider the ethics of CRISPR as the technology was being developed, in relief to the previous discussions about TALENS, whether we'd be sort of in the situation we are now, scrambling to try and figure out how to respond from an ethical perspective to CRISPR-Cas9. One of the major problems that Sankar and Cho note about trying to develop some sort of framework for scientists to use to think about and establish their emerging technologies for social responsibility is that there's just a lack of consensus on what social responsibility is. And as a member of an IRB, I can attest to this, that, there are lots of different opinions on the board about what constitutes societal benefit. And so if we're trying to decide whether or not the risks of a research protocol are justified by the social benefit of the research, you'll get probably lots of different operating definitions of social benefit, such that you could get people who agree on the risk, agree on what the consequences of the research would be for society, and disagree whether or not those consequences are socially beneficial. So that is a definite problem, and vague definitions from institutional bodies or regulatory groups, and in philosophical articles don't really do anything to clear this up. You get things like social responsibility is striving to promote the good of society through your research. And this is, not a new problem. There's a long history of calls for social responsibility in research, and Sankar and Cho suggest that it becomes much more frequent that there are calls for social responsibility in scientific research, and yet we're no closer to getting a definition of social responsibility. This is, I think, sort of also the major shortcoming of Sankar and Cho's article, because I don't think they give us a definition of social responsibility. I think they present to us a framework for what might be responsible conduct of research, but it's not clear that this is going to always establish whether scientists are engaging in socially responsible research. In order to generate a framework for how to conceive of socially responsible research, Sankar and Cho called through researchers' own discussions of what made their research socially responsible, and they have identified 5 categories that scientists often pointed to explain why their research was valuable, or socially responsible. And those are the basis, that is the justifications for the research that scientists offered, and these often invoked values. The approach is the next category, and when scientists justified their research, they pointed to the ways that they identified the harms and benefits of the project to establish that it's socially responsible. The other thing is timing, so when in the development of a research project did scientists address social responsibility? So do they do it at the beginning? Do they do it in the middle? Do they do it at the end? And does pointing to a particular time help to support that it's socially responsible? Like, if you wait till the end to address that question, can it be socially responsible? Next, scientists pointed to who they involved in addressing social responsibility. So who they involved in their reflection on the appropriateness of their research, and that is the category of participants. And then last is the category of transparency. That is how easy it is to identify the ways in which researchers addressed social responsibility. So you got those tables in the article that reflects the development of an H5N1 influenza virus and the development of a gene drive research project using CRISPR-Cas9. And so you can go through there and see how the scientists addressed these categories in their conception of their research. Sorry, I didn't realize the camera was recording just there. So, according to Sankar and Cho, the researchers on the CRISPR-Cas9 gene drive project saw that the basis, or proposed that the basis of their research was to benefit society, and we don't see that in the table. It's just their reports of their, I guess, experience going through the literature. But the other one is that it overcomes a technical problem in science. So they justified this project on the basis of needing to solve a technical problem. If they said they could see benefit to society, it's not clear what that benefit is. And without that, it's hard to assess whether the risks are worth the benefit. So in their approach to establishing the social responsibility, it says that they engaged in a multi-year review to identify potential harms to ecosystems and mechanisms for managing them. So that is a kind of proactive step to make sure that you're not going to create any unforeseen harms. Timing, here they thought about the social responsibility of the research after they got funding for it, but before starting the project. One wonders whether or not you should think about that beforehand. If your project isn't socially responsible, should it get funding? Should you apply for funding? They consulted with interdisciplinary experts on their plan for research, and then they got public consultation on the plan after that. Sankar and Cho's focus, therefore, is on what scientists can do themselves to ensure that their research is socially responsible and perhaps also ethical then, because it's socially responsible. And so they proposed a kind of set of guidelines that researchers are supposed to ask themselves. Now, in Human Germline CRISPR-Cas modification towards a regulatory framework, Niklaus Evatt, Shamik Masharak, and Russ Altman are trying to propose regulations for phasing in CRISPR-Cas9 genome editing therapies that is responsible and doesn't outpace ethical examination. So they are going to propose a regulatory framework to take us from where we are now with the prospect of developing these techniques that requires research to the actual marketing and distribution of techniques for genome editing to potential parents of children who might have whose children might inherit genetic disorders from them. And so one of the things that Evett, Mosharock, and Altman stress is that the only practical policy to adopt towards CRISPR-Cas9 is regulation. You can't put a ban on it. You can't impose a moratorium while we think about the issue more. The fact is that CRISPR-Cas9 is, as I said before, far too inexpensive and too easy to use to really hope to stop people from doing research with it, and the variance in regional ethics codes is too great to guarantee that everybody is going to have a set of regulatory guidelines in their, a set of regional ethics guidelines that are the same. The other thing they say is we can't just leave it laissez-faire, that the risk of the research proceeding without sufficient ethical analysis is too great, and I think the potential implications and consequences of the research is far too great to risk unscrupulous people working in this field. And I think the example of stem cell therapy clinics in countries with poor regulation of medical care is evidence that you can't just ban it in countries where there is robust structure for regulation. I think the other thing is that if you regulate it and encourage responsible and talented scientists to research this with your blessing, you're going to get perhaps better results. So Evett, Mosharock, and Altman think that there are 3 phases of the development of CRISPR-Cas9 that have to be regulated. There's the preclinical research, that is the laboratory research or basic science research that researchers want to engage in to learn more about CRISPR-Cas9 and to see what kind of applications can be developed. Then there are the clinical trials that test the efficacy and safety of those techniques developed in the preclinical research. And then there's the post-approval distribution of CRISPR-Cas9 technologies that once they've, say, gotten FDA approval, how are you going to regulate them? So those are the 3 areas they need, and we'll look at the standards they propose for each now. Evett, Mosharock, and Altman are conceiving of the day when we develop CRISPR-Cas9 gene editing technologies for human embryos. So their regulatory framework is guiding that development. So for the preclinical research, one thing they argue is that no research should be done on changes to DNA that involve gene drives. That is, changing the DNA in such a way that the genes that you change are almost certainly to be inherited by the next generation after the generation you edit, and so on into subsequent generations. If it's possible to do that with the DNA, so Evatt, Mosharock, and Altman say, no, you can't do that. You can't make it so that somebody is more likely to pass on their traits than somebody who is not been edited. They propose that you have to develop reversal mechanisms for the gene editing as you go along, so that if you change a gene, you're able to change it back. They say that there must be clear benefits for patients for this research that you couldn't get without the research. So for instance, if there's prenatal genetic diagnosis or embryo selection for therapeutic benefit for a disease, then you shouldn't do research on it with CRISPR-Cas9. You shouldn't edit the human genome unless you need to. So if you can prevent an embryo from developing into a fetus in a child who has, say, cystic fibrosis, just for example, because you can test the embryo before implantation, then you shouldn't do it. And the diseases that you do research in should be with large potential patient pools, right? So you shouldn't do research into small pools, but a lot of people should be affected. Now, that is a controversial position because it could turn out that a mild genetic disease affects a very large patient pool, whereas a really, really terrible genetic disease only affects a small patient pool. And according to Mosharock and Altman, then that would seem to be a reason against doing research on the terrible disease, because it doesn't hold the prospect of benefiting a lot of people. Next, they propose that you need proof of concept. So one, you need to show that the changes are made on the somatic cell level before you progress to the DNA of an embryo. And you also need to conduct the study in the change of the genome in animal models that breed faster, so you can see the effects of the change over multiple generations, right? So you want to get a sense of how this change will impact future generations as well, and so you need to use animal models. Finally, they propose specialized oversight committees. So IRBs only oversee human subjects research, and embryos are not considered human subjects research. So the authors propose that there should be a specialized committee that oversees the kind of research on embryos that CRISPR-Cas9 research would involve. So this is a kind of interesting position, and I think it depends on, sorry, nothing depends, but that it'd be important to establish what is the responsibility of the oversight committee. Is it to ensure that only good research gets done, so when it moves to the next phase on humans, embryos that are going to be implanted for development into fetuses and children, that you have safe research or safe intervention? Or is it, that we're concerned with the ethics of this kind of CRISPR research itself, and that's why it needs oversight? So are we trying to protect potential future humans, or are we trying to ensure sensible research gets done? So moving on to the clinical trials, Masharak and Evatt and Altman consider what happens when you translate uh, the research from bench to trials that are going to be done on human embryos that will be implanted for gestation and development to birth. So the FDA has a clear set of policies for the use of gene transfer therapy, and the authors proposed that you could incorporate CRISPR-Cas9 into those guidelines as well. Just the same policies should govern CRISPR-Cas9 as govern gene transfer therapy. They also say parental informed consent for first generation participants is necessary. And at first I thought, wow, that's a kind of weird thing to propose. But what I think is in mind, namely, is that you have to get consent from the parents of the embryo that you're going to alter and gestate for this to go forward. So you can't take, I think, discarded embryos and do this and implant them in somebody else without telling the parents what you're going to do. So I think this also suggests that you probably couldn't do this research on created embryos with a volunteer surrogate. Somebody has to be there to parent the child. So whoever's going to be the parent of that child has to consent to this process and taking on any of the possible unforeseen consequences of this. They also proposed that a higher standard of informed consent is necessary. And I'm not sure what a higher standard of informed consent would look like for any kind of trial. I understand that there are different standards of consent when it comes to documentation. Do you need a signed consent? Will something less do? But I think informed consent requires that participants understand that they're getting involved in research, what the research will be require on their part, what are the risks and benefits of participating in the research, who will they be interacting with in the research, what are the possible side effects of participation in the research. But I don't think you can say there's a higher standard. So that's a weird thing, unless what they are going to say is that you need to more closely assess the actual understanding of the parents. In that case, I think you'll run into issues of, just whether or not you should sort of paternalistic keep checking understanding. And finally, they want ongoing studies after approval. So once these are therapies that are in use in clinical practice, they want ongoing observation of the embryos that were created from the CRISPR-Cas9 interventions, but then also their descendants to keep an eye out if there are any changes or effects that we should know about. Finally, they consider the regulatory framework for distribution of these kinds of therapies. So specifically, they express a concern that people who could benefit from these therapies, people who will pass on inheritable conditions to their children and would like to have genetically related children will not be able to access them because they can't afford access to IVF. So you might know, for instance, that you or your partner are carriers for diseases that one of you can transmit to your child, or both of you could transmit together because you're both carriers to your child, through genetic testing. So if you were proactive before trying to start a family, say, and you got genetically tested, you could find out that to a high degree of certainty whether or not your child was at risk for contracting or inheriting the genes that would cause a genetic disorder, and not be able to do anything about it because you can't afford in vitro fertilization to do, say, PGD, or even, to do it at all. So if it came along that there was CRISPR-Cas9 technology to transform embryos, and you couldn't afford it, Ebbett, Masharak, and Altman are concerned that that would be an unfair situation. The other concern they express is that insurance companies may raise premiums on subscribers who don't use CRISPR-Cas9. So if I have a child, if I'm a carrier for a disease and my wife is a carrier for a disease, and we're going to have a child, and we choose not to use CRISPR-Cas9 to make sure the child doesn't have the disease, insurers might want to say, you have to pay a higher premium because of the fact that you didn't avoid passing this expensive to treat disease onto your child, and you should therefore pay a higher premium. The authors think that would be unethical, and I think there's every reason to agree.